Welcome to this video on the kidney dissection. This is a sheep kidney which we use for its similarity to human organs. You can see that it's surrounded by a fair amount of fat and then a kind of a whitish membrane around the outside called the capsule. And I'm just removing the fat here to expose the kidney proper. Somewhere in there is probably the adrenal gland. And the white connective tissue is mainly a protein called collagen. Okay, I'm just going to start off by putting the kidney on its kind of inner edge and cutting down through the outer long edge of the kidney in order to expose the layers inside. And we're going to go in very slowly, kind of layer by layer. So the first layer that we can see is kind of a brownish colour, and this is a layer called the cortex, C-O-R-T-E-X. And as I go in a little bit deeper with a scalpel, you start to see a kind of more pinky red layer appearing, which is the medulla. Now, starting to appear through the pinkness of the medulla, I can see another even paler layer, and as I go through the last little bit of medulla, we'll see down into the white inner side of the kidney, which is called the pelvis. Not to be confused with your hip bone, this is a different pelvis. So with the kidney laid out flat, we can clearly see the outer cortex, the sort of central pinkish medulla, and then the white inner pelvis here. And the edge of the pelvis forms these kind of pointed structures reaching up in towards the medulla, and they're called the calyces. And then the pinkish medulla coming down, forming pointed structures back down towards the centre. Those are known as the renal pyramids. I'm just showing them here. Just continuing to clear away bits of medulla here, so you can see those white calyces pointing up from the pelvis and the pink medulla pyramids pointing down the other way. Those pyramids are made up of many thousands of tiny tubes, and urine flows down those tubes in towards the centre, and eventually it goes out through this hole, which is the opening to the ureter, which is the tube that leads to the bladder. So it comes out the other side, and you can see what's left of it here. It hasn't survived being removed from the sheet very well. Now I'm just going to add a little bit of hydrogen peroxide solution to this. Now hydrogen peroxide is broken down by an enzyme in the kidney tissue, and it produces lots of tiny little oxygen bubbles. You can see the oxygen being produced. Those bubbles get stuck inside the tiny tubes of the kidney, and it makes it easier to kind of visualize them, sort of. It's easier to see this live. If I just complete the cut now all the way through, because remember I've cut in from the back edge, so completing the cut all the way through the kidney, you can kind of see the original kidney shape more clearly now. So that's the ureter in the centre, surrounded by the pelvis and the pinkish medulla, and the brown outer layers, the cortex.